Hello, so welcome to our next section of this chapter in regards to energy. In the last video, we talked about photosynthesis. I'm going to tell you now that this information can be a little bit overwhelming. I will provide other resources in order to hopefully get that information to you in a different way so that you're not as confused. So remember when we talked about photosynthesis, we were converting light energy and making glucose. Now we are talking about how living things use this energy in the form of cellular respiration. After this video, you should be able to name the 3 stages of cellular respiration, give an overview of glycolysis, explain why glycolysis probably evolved before the other stages of aerobic respiration, describe the mitochondrion and its role in aerobic respiration, list the steps of the Krebs cycle and identify its products, explain how electron transport results in many molecules of ATP, and finally state the possible number of ATP molecules that can result from the aerobic respiration. So a lot of things we're covering in this video. It may be a little bit lengthier than usual. I apologize. I am going to try and make this as quick as possible. We will go over all of this in class. Please don't freak out yet. All right, so stage 1, or the stages of cellular respiration. Here is a nice picture showing all 3 of those stages. The first stage is known as glycolysis, the second stage is known as the Krebs cycle, and the third stage is known as electron transport. So that word, the electron transport, should sound familiar to you from photosynthesis. So that stage is going to be stuff you pretty well already know. We're just maybe using our different characters or players in different ways. Big thing I want you to notice with is, remember, we start with glucose. That is what was made, and that's one of the things we use when we're talking about cellular respiration. That's one of our reactants, if you will. We're going to break those up in glycolysis. So stage 1, we're breaking up glucose to create 2 pyruvate molecules. In stage 2, we take those pyruvates and we expel out CO2. We also make some NADH, FADH2, and ATPs. Those go on, NADH and FADH2 go on to the electron transport chain, where we make some ATP. So that's a brief synopsis of the stages of cellular respiration. I'm going to go into details in the next few slides. The first stage of cellular respiration is known as glycolysis, and this occurs in the cytosol of the cytoplasm. So in a regular animal cell or a living organism, so we're not talking about plants anymore, we're talking about living organisms, it occurs in the cytosol. So basically what glycolysis is, it's splitting glucose. So remember, we start with glucose. We are taking that 6-carbon chain and we are breaking it up into smaller pieces, because glucose by itself is just way too big for the cell to use. So it breaks it down and creates some really nice usable energy. So how this occurs is we have specialized enzymes. Remember, enzymes are just proteins and they do specific jobs. Enzymes, they split a molecule of glucose into 2 molecules of pyruvate, also known as pyruvic acid. I will probably always just call it pyruvate. So they take the glucose, split it up into 2 pyruvates, which are just 2 3-carbon chains. So during this process of splitting glucose, it always requires energy. So there is going to be energy that is absorbed and also released in this particular stage. So when this occurs, we started with glucose. We now have 2 pyruvates, which are 2 3-carbon molecules. Those pyruvates are going to move on to stage 2. But we have now we've released some energy from that reaction. That released energy allows for the making of 4 ATP molecules. So we used that form of energy to split the glucose, but we are now also making some. So we have a total net gain of 2 ATP during glycolysis. So it may not seem like a lot, but that ATP goes on to help contribute to a huge number of energy source for the entire cell. These excited electrons are transferred to NAD plus to produce NADH. So remember, in photosynthesis, it was NADPH. Please don't confuse that with cellular respiration, which is NADH. NADH is used in stage 3 to make more ATP. All right, before we progress on to the next stages, we need to talk about some different types of respiration. One is known as anaerobic, one is known as aerobic. And this involves glycolysis because of the fact that glycolysis is theorized to be more or to have evolved before the other stages of cellular respiration. So millions of years ago, when the Earth was formed, there was no oxygen in the air. Oxygen did not begin to come into the atmosphere until after organisms that could undergo photosynthesis were created or were born, if you will. So that's why glycolysis is one of those stages where oxygen is not required. So all of the other stages in cellular respiration require oxygen. Glycolysis does not. So and that's obviously a lot to do with the Earth's atmosphere at the time that evolution occurred. Cellular respiration that proceeds without oxygen is called anaerobic respiration, meaning I do not need oxygen for this to happen. So glycolysis is a form of anaerobic respiration. After a progression of evolution occurred and with photosynthetic bacteria being formed, that was when oxygen was introduced to the atmosphere and when the entire cellular respiration or that evolution of structure became a thing. And so aerobic respiration, a more primitive form of respiration, is respiration with oxygen. And those are two important terms you definitely should know. Like I said, all other stages of cellular respiration require oxygen. Glycolysis does not. All right, and another thing you do need to know about before we progress on to the next two stages is the structure of the mitochondrion. You've also heard me call it the mitochondria. And so everything in regards to cellular respiration the next two stages occur in the mitochondrion. So remember, glycolysis occurs outside of that. Inside of the mitochondrion is where we now involve oxygen, and we are undergoing the next two stages. A couple of things you need to know. The mitochondria has an inner and outer layer, and that's really important because those two stages occur within the inner and outer layer and the intermembrane space. The inner membrane space is located between the inner and outer membrane. And I know some of you are probably thinking, yeah, that's obvious, but I still need to say it. Within that inner membrane, we have what's known as a matrix. And this is where the Krebs cycle is going to take place. The third stage, the electron transport stage, occurs in the inner membrane. So we're going from the matrix out of the matrix, and that's where stage 3 occurs. All right, the next stage known as the Krebs cycle. This is where we use those two pyruvate molecules that we got from breaking down glucose. So remember, in glycolysis, we broke down glucose and we created pyruvate. So 2-pyruvate. Pyruvates, they enter the matrix. That's where the Krebs cycle occurs. Before this actually happens, there are things that happen to the pyruvate. So pyruvates are split even further, and they combine with coenzyme A. So CoA, producing acetyl-CoA. So there's a few things that are going on. Pyruvate split, so one of them will split. Both of them will split in the process. It repeats a couple of times. But pyruvate splits, it combines with CoA, producing acetyl-CoA. What you have left over is carbon combines with oxygen to release CO2 as a waste product. So again, that happens all before the Krebs cycle actually begins. Acetyl-CoA, so now this is the beginning of the actual Krebs cycle. So let me actually star that really quick. This one here is the beginning of the Krebs cycle. Everything before that, or everything happens before the Krebs cycle that I've talked about so far. So acetyl-CoA combines with a nice fun guy. So this is a 4-carbon molecule called OAA, oxaloacetate. So just call it OAA. And this produces citric acid, which has 6 carbon atoms in it. This is why the Krebs cycle is often called the citric acid cycle. I don't expect you to know that, but I do expect you to know that acetyl-CoA combines with OAA. And those two things combining form citric acid. After citric acid is formed, it goes through a series of reactions that release energy. That released energy is captured in molecules of NADH, ATP, and FADH2. So all of these things are just helping energy, and they're captured, and they're helping move that energy. Like I said, carbon dioxide is released due to the just added up carbon and oxygen that are present. The final step of the Krebs cycle regenerates OAA, the molecule that began the Krebs cycle. So again, this is a cycle. It repeats itself. This molecule is needed for the next turn through the cycle. There are two turns that are involved in here because remember, glycolysis produced 2 pyruvic acids. So 1 pyruvate goes through this, the second pyruvate goes through this. The results after all of this has happened is that you have 4 ATP, 2 from glycolysis, so you really created 2 from the Krebs cycle, 10 NADH, 2 are from glycolysis, and 2 FADH2s. I can already feel your confusion. It's okay. We're going to go through this in class. The final stage of cellular respiration is known as the electron transport stage. And so again, final stage. In this stage, energy from NADH and FADH2, which were the results from the Krebs cycle, is transferred to ATP. So that energy from those 2 molecules is transferred to ATP. And how does this happen? So really the biggest hint I can give you is remember the electron transport chain in photosynthesis and how did that occur? It's a similar process. So high energy electrons are released from NADH and FADH2. Quick reminder, NADH and FADH2 are energy carrying molecules. They carry around energy. So they're going to release that energy as they move along a transport chain. Just exactly like in photosynthesis. The electron transport chains are on the inner membrane of the mitochondria. So we're now in the inner membrane. So we've gone from the matrix, we're in the inner membrane. As the high energy electrons are transported along the chains, some of that energy is captured. The energy is used to pump hydrogen ions from your NADH and your FADH2 across the inner membrane, from the matrix into the inner membrane space. So we are going from the matrix to the inner membrane. The pumping of hydrogen ions across the inner membrane creates a greater concentration of ions in the inner membrane space than in the matrix. So we don't like that. So we're always going to go from high to low. So now we have what's known as the chemoosmotic gradient. Probably sounds familiar. You have the high concentration of hydrogens. You want to pump them out. So the chemoosmotic gradient using ATP synthase allows that flow of those H pluses. Now, ATP synthase, again, acts like a channel protein, helping hydrogen across, but it also acts as an enzyme forming ATP from ADP, exactly like it did in photosynthesis. So chemoosmotic gradient uses ATP synthase, not only moves those hydrogens across, it also creates ATP. At the end of all of this process, you have a huge number of ATPs created, and that's what we want to do. We went from glucose to energy. Glucose to energy. And how much ATP do you really think we have? Well, here's our number count. 2 in glycolysis, 2 in the Krebs cycle. In the electron transport chain alone, you get 34. That's a huge number. So a total of 38 ATPs are created from cellular respiration. 1 thing of sugar, and we're eating tons of sugar every day. Everything we eat has glucose in it. So this is really an effective process. We get enough energy. That's why it's not a good idea to cut things that you eat. Now, I know that this information was super confusing and you may be really confused. We are going to review this. Do not worry. You will have the tools you need to succeed. If you have any questions, please ask me on Edpuzzle.